So next week will be the student presentation, and we will have, I'm thinking about 15 minutes per group, and I hope all three of you in the same group will equally distribute that share. And I'm going to tell you the order of the presentation of different groups maybe early next week. After that, we will have, I think, that will be the Thanksgiving break. And after that, December 2nd will be our final exam. And today, the lecture will be two parts. The first part, I will be very quickly go through the epigenetics part on the ChIP-seq. And the second part, I'm going to give, I think we're good. Yeah, thank you. And on the entire course evaluate, the knowledge points that I want to give a brief review on that. If you have any question, and that's a good time to ask. So let's get started from the ChIP-seq. This is the third lecture of the ChIP-seq. So last time, we talked about the experimental considerations for the ChIP-seq. The first point is antibody is important. We all know that now. And then the initial material, the total amount is very low, so there's some special considerations on that. And because of this, they are pretty low, that we have the resource actually to barcode several different samples into one sequencing run or into one lane of the Illumina platform. And sometimes people consider pair ends, and that will help to analyze areas in the repetitive regions. If you study a transcription factor, it may not be very useful sometimes. But if you are studying some chromatin markers, that is more preferably in the repetitive regions, it probably will help you a lot. And we also talk about the sequencing depth evaluation. There are two major approaches. The first one is the saturation analysis. That will be after the experiment, after the preliminary experiment, so you kind of can evaluate how much more sequencing resources do I need to get the more information figured out. And there are also an equation that, if you recall, that talk about the evaluation before the experiment. That's an estimation. That is basically depends on the genome size, the window size, or the resolution you're interested in, and how significant that you want the eventual results are. And we also talk about the fragmentation approaches. There's two basic types. One is the enzyme-based, and the other one is the physical breaking it. So it's a physical shearing. And the good thing of enzyme-based is it can give you down to the resolution of one nucleosome. And but the problem for the enzyme-based is sometimes it has a certain sequence specificity if you use enzyme to fragment your DNA. And the last point for the experimental consideration is the controls. So we talk about there's three different types of controls. You can go back to your notes to look at that. And it's IgG control and no input control and things like that. And we also talk about it's possible that you don't need any control if you're considering the comparison of two different conditions. And the second major point is when we talk about ChIP-seq, we always think of it's a transcription factor binding sites. And if that's the transcription factor binding sites, the signal distribution will be more look like the first one. And you can see there's a cluster in the positive strand and in the negative strand, and they are kind of shifted because of, so we will have next slide to talk about why that happens. But for the second part, there's a RNA polymerase II. So you can see there's a gene expression. This is a transcription starting site. And around promoter region, you will see this nice easy peak. And then after that, there will be transcript regions, and you can still see reasonable enrichment. And then by the end, the Pol-II pausing and eventually get off from there. And for the third part, which is epigenetic markers, so you can see this is a longer range of that. There could be several thousand base pair, and you see a gradually enriched signals along there. For the last lecture, we talked about this data type for the first type, which is the transcription factor binding sites. And today we will talk about more of the information on the second two classes, the second and third classes. And in terms of the first one, which is the transcription factor binding sites, and we know that, so we spend a little bit of time to talk about what the data distribution will look like. So you can see this is the protein, and this is a double-stranded DNA. And the red one is on the positive strand, the blue one is on the negative strand. Because we are sequencing this DNA always from 5' end to 3' end, so you can see there will be significant enrichment on the red part on the left side of the protein binding, and the blue part on the right side of protein binding. So when you really put the signals together, it looks more like this. And there's two separate peaks of tag density on positive and negative strands. And the distance between these two peaks actually reflects the size of a protected region, which is the size of the protein binding sites, and also influenced by the size distribution of the DNA fragments. And when we identify the peaks, there's generally two different strategies. The first strategy is called the tag shifting strategy. So the principle is very simple. It just you got all the positive peaks and negative peaks, and they just shifted towards the inward side for half of the estimated average DNA fragment size. And we have later slides talk about how do we identify the DNA fragment size. But once you have the DNA fragment size identified, you just shift your signals towards the inward for about half of that size. And then you will see this nice converged peak that will tell you almost exactly where the binding occurred. And sometimes you want to do this for both, if you have the control signals available, experiment available. This is a chip-based experiment. This is a control experiment. So you can see in this region, you can still do that type of shifting, but eventually the overall signals will be very flat. So, and then at this stage, you're comparing this derived tag-shifted signal and versus the control. And then you look at this enrichment, and then this is the way to help you to identify where the binding occurred. We're still OK? And this is basically the first approach, tag shifting. And the second one is tag extension. And there's some algorithm that is using this type of approach. So basically, it's very simple. It's just because what we measured is only the very beginning of each individual read. And then what you do instead of you shift it, rather you extend it and make it to the original length, by average DNA fragment length. And then you will be able to get more signals in this restricted area. And for this type of approach, the advantage of that is, number one, it corrected for the assumed fragment length. If it's 200 base pair, and then instead of just the 36 of the sequencing read length, you extend it to the entire DNA fragment length. And also, it's smoothing over gaps. So for some region that you may, because of the short reads feature, it may not be mappable. Remember when we talked about DNA sequencing part at the very beginning, we talked about mappability. Sometimes it may not be mappable. If it's not mappable for that particular region, you probably don't see any signal there. But when we do this tag extension, and that part will be fixed, the gap has been glued together. And so two different approaches, tag shifting and tag extension. And another thing that we talk about briefly is the sliding window strategy. So most people, when they do DNA sequencing, ChIP sequencing analysis, they do not use one individual nucleotide as a base, as a unit. Rather, you use the window size. That window size can be 100 base pair, can be 200 base pair, can be 20 base pair. It depends on the resolution that you eventually want to achieve. And but the point here is, once you take that approach, you can see that this is the ones that based on the one base pair resolution. So you can see this is each y-axis responding how many sequencing reads that cover that particular region. And you can see this is kind of very spiky type of profile. But once you look at only a sliding window strategy, and then your data will be much more smooth. And this is the way that to do the denoise. And also, if you take this approach, you have more statistical tools that you can use. For example, for that particular window size, the number of rays fall into that region, you can assume Poisson distribution or negative polynomial distribution. So you have more statistical tools can help you to figure it out. And we also mentioned that this whole thing that we are assuming that this binding site is one independent binding site. There's no surrounding the same protein binding sites occurred. So that's why that we always assuming this type of two peaks and one in each strand. But sometimes we also, if the multiple binding site happens, and there's a possibility that you will see these broader peaks if there's a multiple binding site in this region. And so once you do the analysis, you want to go back to the genome browser to take a further look at whether your strategy is reasonable or not. And I haven't seen any established approach to tackle this problem, but it's very possible that before you discard this type of signals and you want to look for the motif, the sequence features within this region, if you see several things that they are pretty similar and they occurred multiple times in this broader region, that is the indication that potentially there's more binding sites in this region. Is that okay? All right. And so those are the peak finding algorithm and so on and so forth. But there's one important thing that we talk about, we are using in the both tag shifting and tag extension approach is the DNA fragment length. And so people try to decide that initially just experimentally. So they got the gel pictures and they run those the gel, and then they cut the band, and they kind of decide what's the range of the DNA fragment that has been used. But that is not really perfect because you don't really get the distribution of things. And we also mentioned that later PCR steps may favor shorter fragments rather than long fragments. So what you derive, the ones that in the final sample probably is not the same as what you see on the gel picture. So people decided to do that computationally. And this is a very standard approach. So what people do is they calculate the cross correlation of positive and negative strand tag density. And after shifting towards the right direction, for example, if we shift for one base pair and the correlation looks like this, and here in this window, I think once you shift it 100 base pair, you can see the highest correlation between the positive and negative signals. And that's a good indication that most of your fragments at least peak at about 100 base pair. Okay? And we mentioned that there are maybe a dozen, maybe 2 dozen peak finding algorithm has been reported over the past couple of years, especially in 2008 and 2009, because the ChIP-seq kind of came out early, and this is the first the real application that occurred in 2007. And so there's so many different approaches, but which one to really pick? And this is one paper published last year in trying to evaluate the different approaches. And I'm not going to go detail into the evaluation results, but keep in mind at the time that you select the algorithm to use, you don't have to write your own program, but when you pick and try to go back to the paper and to see which one fits your strategy. And after the ChIP-seq data analysis, we talk about, all the previous effort is trying to, you got the ChIP-seq experiment and trying to see where the binding site occurred, okay? Just based on the signal processing, just look for the experimental signals. But after you identify the regions that the binding occurred, you want to understand a little bit more about the features of this protein binding. You want, for example, you got a transcription factor, let's say AP1. I want to know AP1, the binding site, what the site is, what the motif is. And we spend almost an hour of time to talk about motif finding theories, and some of them are not really related to next generation sequencing. So you can see there's a couple of different representations of motifs, and we talk about just the straight letters, A, C, C, G, T, or whatever like that. And we talk about consensus type of motif. But the more accurate characterization comes from this type, which is a position weight matrix, which tells you that for each different nucleotide, the percentage A occurred, C, G, T occurred. So what's the percentage that those different things occurred? And this is called the position weight matrices, and it sounds like a pretty complicated, and you may just initially think, where I can find those information. Actually, they are very well documented for this position weight matrix. We have almost 1,000 different binding sites, and they are very accurate position weight matrices. And we also talk about, after this, we can transfer this to something called the position-specific scoring matrices, and that is just another way of calculating things. And once we got a new sequence, like a 6-base pair, and then based on this position weight matrix, we just check the numbers inside and add them together, and that gives you a calculation for the matching scores. And for the motif finding, there's two major strategies. The first strategy is the de novo motif finding. And so this is, you can see that these are the ChIP-seq derived fragments, but there are something that in common in these regions. Those are the parts we want to identify. And if you haven't done this before, and I'll tell you, this is very, very complicated, because you don't know where they are, you don't know what the sequence features are, and most importantly, they are degenerate. So they are not look exactly the same. This is just roughly the same. So there's many different algorithms around before the ChIP-seq experiment has been developed, and especially around 2005. And there's a Nature paper, I think, biotechnology paper, review paper, comparing different approaches. But those are the strategy that you can do the de novo motif findings. For me, the second strategy makes more sense, and it's easier to do, and then you can design follow-up experiment on those, which is searching for the existing databases, like TransFact database. As I mentioned, there's almost 1,000 binding motifs for the transcription factors has already been documented. And in terms of a motif finding, and this is how we do that, and we calculate for each individual ChIP-seq derived sequences, we calculate the highest matching score, and then we also select the random selected regions, and hypothetically there's not too much enrichment of the binding, and then you also calculate the highest matching score. So, and then you shift your cutoff, and eventually you calculate if my cutoff is 1, and the percentage of random divided by percentage of ChIP-derived signals is 0.88. That's not very good. The false discovery rate is almost 88%. But once you increase the cutoff scores, you can see clearly that this false discovery rate dropped to about 30%. And of course, you can decide what is the cutoff you use to use this type of approach. So if you don't know what I'm talking about, go back to review your lectures, and shouldn't be very hard. And this is one of the examples that in the paper we published that we used the ChIP at that time. I think it was 2007, around that, and identified the estrogen receptor alpha binding sites using ChIP experiment. And we found so many different regions, and thousands of those, and we search for the motif enrichment in those regions. As you can see, these are the factors that eventually show the significant enrichment, and the bar here is about 20% FDR, and these are the different motifs. And here is the real motifs, and then you can see the sequence similarities in these regions. Okay? So last time, basically, we really talk about is the peak finders. Those are the analysis that's specific for transcription factor binding, and also the motif findings. Those are the two things that we talk about. Any questions so far? No? All right, good. So what I'm going to talk about today is the first one is one story regarding the microRNA, and which actually is an application of the ChIP-seq data on the RNA polymerase II. And so we mentioned there's 3 different classes of signals. the transcription factors, the RNA polymerase, and histone markers. And this is the second class, which is the RNA polymerase II related story. And then I will jump into epigenetic regulation. And I think the microRNA part also fits into this because some people consider microRNA regulation as the epigenetic regulation as well. For this part, actually, most of the computational approach are not really well organized at the moment. And it's just specific applications derived by this group, that group, but there's no general theories available so far. What I'm going to give you here is I will give you the basic concept of epigenetic regulation, including histone modification and DNA methylation. And for the histone modification part, I will tell you one approach. Actually, it's only one slide to tell you how we do the data analysis on the non-peak finders. And actually, that's the only tool I have found so far. And it was published in 2009. And I also asked my colleagues from Ohio State and the two professors there, and they even didn't know this one. So you can see that this is still a research area, that computational part that's still yet to be discovered. And for the DNA methylation part, and the analysis tool is even less. And but I'm going to tell you several different experimental approaches using next-generation sequencing. Basically, this will be focused on the experimental part. Okay, so let's first go to the RNA polymerase II story. And this is, if you attended biochemistry seminars that I gave, and this is a short version of that. So what happens is we want to study the microRNA-mediated regulatory network. This is not somewhere we find out from other publications. This is our work, okay? And we want to study the microRNA-mediated regulatory network based on the RNA polymerase II derived ChIP-seq signals. And these are the two papers, if you're interested, and you can go into look at. So very briefly, talk about microRNA regulation. Is there any way that I can dim the light? Probably not. If I dim the light, everybody just automatically goes to sleep. So better make it bright. So you can see that this is the cell, and this is in the nucleus, and the microRNA actually, it is a gene on the DNA, and it will be first transcribed by RNA polymerase II or RNA polymerase III, okay? And about 70% was transcribed by RNA polymerase II, which is the same as protein coding genes. And it has three different forms. So what that means is after transcription, it first generates a very long form, which is called the primary microRNA. And that primary microRNA can be a couple of thousand nucleotides long. And after that, you do this cleavage and form this hairpin loop and get rid of things you don't need. And you form this called precursor microRNA. This is about 100 base pair long. It's a 60 to 100. And at this stage, it's called a precursor microRNA. It will be shipped out of cytoplasma into cytoplasma and further processed. We know that those disers and the androsa, and they eventually become this mature microRNA that is only 22 base pair long. So it's three different forms: primary, very long; precursor, about 100 base pair; and the mature, which is very short. And this microRNA is self-mated, and we have almost 1,000 in human genome. And we know that about 2/3 of the human genes are actually microRNA targets. And so far, what we know of the function of microRNA is mRNA cleavage. So it will bind on the 3' UTR of the gene, and then use other proteins to kill the target RNA, or translational repression and mRNA deadenylation. But regardless of what individual factors are, you can consider microRNA as the brakes, right? So once you make too many, and they start to help you to reduce the speed a little bit further. And I think there's a recent discovery is the microRNA cleavage is the primary form of how microRNA really works. Okay. And our initial goal here is if this is we know that microRNA will target the gene and bind in the 3' UTR of the gene, and then further kill that gene. So microRNA has this kind of relationship, regulatory relationship to the gene. But if this gene is a transcription factor, it will potentially come back to regulate the transcriptional regulation of the microRNA. So if this happens, we are hoping that we can form this kind of network to bring those information back together. And initially, we have this very interesting data, and that was in collaboration also with Harinakashatri's group. And we had both gene expression data and the microRNA, microarray data for the same group of cells. So we thought that we can bridge everything together and make a network like this. So different components that interacting with each other and how that will work, okay? But the data itself, the network itself, it really never worked. And we can always form this type of network, and I almost feel I need a GPS to really read this type of network, and the results never made sense. And I think the reason for that is at that time, when we try to build a network, we don't understand the microRNA biogenesis very well. So here's the problem, okay? And I mentioned there's 3 different forms of microRNA: primary, precursor, and mature microRNA. And this is, if I put that on the DNA, this is the primary microRNA. It's very long, and precursor and mature, right? And if we think of study how these microRNAs are regulated, and we better go to the transcription starting site of this microRNA and look for the promoter regions, and further trying to look for which transcription factor is binding in those regions. And so we better know the promoter regions of this microRNA. But actually, when we go to the microRNA database, what we got is always this part, which is where the start and end of the precursor or mature microRNA. So we never had the primary form where the start is. And there's no database on that. People don't know that very well. So when we look for the binding sites and of transcription factors we're interested in, we always go to this region, which is really off the target. It's not on the promoter regions. So our goal for this project is at the time that we have RNA polymerase II ChIP-seq data, and we were thinking whether we can identify the regulatory regions and transcription starting site, identify these just based on ChIP-seq derived RNA polymerase II binding patterns. So that's our initial goal. The strategies we use is actually very simple. We got the ChIP-seq data on RNA polymerase II, okay? And we want to really learn from the protein coding genes. The reason for that is for these protein coding genes, we know, at least we think we know, where this gene starts. So where the transcription starting site is and where the promoter region locates. And for these genes, and we can really look for the RNA polymerase II binding patterns around this region and trying to find out is there any common features of the binding patterns. If we were able to figure that out, we can further apply that on the microRNA because we know where the microRNA is, and then we just go upstream to search for the similar patterns and to further identify the promoters. So this is our general strategy. And we got 2 papers on that, and which is published last year. And so let me show you some results. So the first step is, again, we want to learn from the protein coding genes to see whether there are some specific binding patterns around the transcription starting site. So the goal here is to confirm whether RNA polymerase II binding pattern follows certain patterns for high and low expressed genes. If there is a clear pattern, what does it look like? Okay, and the data we use is we have the same in the same cell. We have the Affymetrix microarray data. So we just use a very crude classification, absent and present. If you work on the Affy data, you know that absent means those genes are really low expressed, and present means that at least you can detect their signals with arrays. So here is the distinction. For the genes that express when we can detect in the arrays, the RNA polymerase II ChIP-seq signals look like this, okay? And for the ones that we were not able to detect in the microarray, and it looks like this. Okay, so you can see, still see a small bump here, but it's not really too much. And if we further subclassify these the genes expressed into different categories based on their expression levels, you can see the higher the expression levels, the more signals you will detect in those regions. At this stage, there's nothing surprising at all, right? This is exactly what we thought, and this is something that we want to really learn from. So let's go back to this initial, the pattern we look at a little bit further. So what we see the key features. The first one is we will see a spike around the transcription starting site in this region, and which is not surprising. And in both promoter regions, which is upstream, and downstream, which is gene transcript regions, we see this gradually goes down, the signals. This is dropping. And we also see that in the transcript region, in this region, the signals is higher than in the background region. Again, nothing surprising. We just want to look this in a little more detail. That can help us to design some graphic models to model this whole shape. So we will, let me see whether the next slide, whether we have this. All right, let's go back. We'll spend the rest of the class to talk about that equations, but let's first look at this. And so basically, we just use several parameters to model this shape. And the first one is the signals around the transcription starting site, how big this signal is. And then the signals around the background region and in the transcript region. So 3 signal intensities. And also the decay rate. There's a 2 decay rate. How fast the signal decays in the promoter region here, and how fast it is in the transcript region. So these 5 parameters help us to model the entire shape. And then we divided this region into multiple bins, and each bin is about 200 base pair. And we want to see that using a Poisson model to model the whole process. Okay? If anyone has interest in the equation part, we can talk about it offline. And I'm not going to talk about it here. All right. So, but we may have a quiz after this and to just do this equation to solve those. All right. Probably not, so don't worry about that. And let me give you the direct application and show you the results. So the data we are using here is a published data, published in Nature Genetics, Biotechnology, and this is on the STAT1 ChIP-seq data in HeLa cells. And after, so they have the ChIP-seq data on RNA polymerase II ChIP-seq, RNA polymerase II in HeLa cells, and also on a transcription factor. So you may see this type of peak finders, and using STAT1 and in interferon gamma stimulated HeLa cells. So these are the data we are using. And in the summary of the results, so we were able to, using this strategy, identify 83 active microRNA promoters from 419 intergenic microRNA regions. We only focus on the intergenic microRNAs, meaning that there are half of the microRNAs there actually in the intron region of the gene, and most likely they will share the promoter with the host gene. So we don't want to mess up with that. Although earlier this week I went to Case Western, gave a talk on this work, and one of the question was there are some of the gene microRNAs even there are in the promoter in the intronic region of another gene. They may have independent promoters and not using the host gene promoters. And actually, we did this. And one of the reviewers of the paper also asked the same question. And most of them, so our observation is about 90% of the intronic microRNAs, they share the same promoter with the host gene. But we also see about 10% they are not. And some of them really stretching far, far away, about 30,000 base pair upstream. And it has been confirmed they are really the promoter of that microRNA. But those are sad stories. But here, we only focus on the intergenic microRNAs. And we found 83 of them that we do see the PAL2 enrichment. And then we, of course, we need to do some certain level validation. The first validation is for the regions we identified, whether we see the conservation, whether the region we identified, they are evolutionarily conserved, and our conclusion is it is yes. And the second one is we want to look for the CPG island, because we know the promoter region, about 60 to 70% of the promoter regions, they actually overlapping with the CPG island, which we'll talk about CPG island in the second part of this lecture. And we found that for the regions we identified, 71% actually overlapping with the CPG island. This is a good indication. And the third one is we look for the histone 3K4 dimethylation markers in the region we identified. This is the transcription starting site. And then you can see the shape looks like this. And this is the Cell paper in 2007, and identified the promoter region of the protein coding genes. The same histone marker, the shape looks like this. So this is almost identical with what we have seen based on the computational approach we use. So you can, this is another level of confirmation that the region we identified is correct. And this comes to the application of what we talked about in the last lecture, the motif finding part. And we identified these promoters, and then we want to search whether for the binding sites for STAT1 and other cofactors. So we want to really look for the binding sites of the region we identified, and whether it's a STAT1, and whether there are some collaborating factors in those regions. Okay, so STAT1 binding sites has 3 different potential models. The first one is STAT1 binds in the promoter and do the regulation. The second one is STAT1 actually sitting on top of another factor. And the third one is doing like this. And in our data, so we want to see from our data which model really worked. So you can see if this happens, we will see a STAT1 binding site in the promoter region. If this happens, we will not see STAT1 binding site in the region we identified. Rather, we will see the binding sites of another factor, which we don't know at this stage. That's the point of a motif finding part. And if this happens, we will see both. So we want to see whether both of the STAT1 and another factor will be enriched in the region we identified. And we want to see what's going on there. And after that, we want to summarize the regulatory network. OK, let me show you the motif finding part. So we found that 41 out of 83 promoters overlapping with ChIP-seq derived STAT1 enriched regions. And we just did this motif search in these 41 regions. And the first motif, using exactly the approach that I showed the last time. Okay? And then we found out the first motif showed up was a STAT1 binding site, which is kind of nice, right? And then we also found out the enrichment of many different other motifs in these regions. And these including the AP1 and a couple of others. But if you look at these patterns more closely, you will find out that they have very big similarity in terms of their sequencing features. And that really suggested one of them is real. The others may be artifacts. So we think AP1 is real at this stage. I'm not going to tell you what's the reason for that. And but another thing that we look at is we found out among all these ones that has a STAT1 binding sites, we had detected 37 of those. And 19 has AP1 binding sites in those regions as well. Okay, so what this really suggested is both these 2 models worked. Remember, there's another one that there's a cofactor in the STAT1 sitting on top of it. We haven't seen that. If that occurred, we should be able to see the AP1 binding sites and in those regions and no STAT1 binding sites. But eventually, what we see is all the ones that AP1 is enriched in there, we have STAT1. This really suggests that these 2 different type of model worked. So you can see, I'm just trying to show you that using the approach that we talked about last time, that you can really, if you use those creatively, that can help you to answer some of the biological questions you're interested in. So after having all those hassles, identify the promoters, identify the motifs. So our goal is looking for the regulatory networks. Again, if this microRNA regulated gene, if this transcription factor will come back. So what we really see is interferon gamma, we know it will activate STAT1, and STAT1 is in the promoter region binding of these microRNAs, and this microRNA can potentially come back to kill interferon gamma. This is a so-called negative control, and that is one of the mechanisms to keep the cell in shape, right? If you have something outside and triggered interferon gamma, and the cell has its own response and trying to balance this to kill the interferon gamma at the same time. And this is another type of pattern we have seen, which is interferon gamma stimulates STAT1. STAT1 also regulates these two AP1 binding proteins, and it also both AP1 binding protein and STAT1 are in the promoter region of this microRNA. And this microRNA will come back to kill both these two AP1 binding proteins and kill interferon gamma. Okay? So, and the third pattern is this feedback relationship comes to the STAT1 and or only STAT1, no interferon gamma, and all only AP1 binding proteins, and no STAT1, no interferon gamma. So putting all these things together, that we will be able to make this a more meaningful network, which is interferon gamma activated STAT1, and STAT1 controls these microRNAs, and then some microRNA will come back to regulate the interferon gamma. Others will control STAT1, AP1, and the interferon gamma. So you can see this type of network that can give biologists more choice to design further experiment in their study. So the summary of this part is the genome-wide RNA polymerase II binding patterns from ChIP-seq is a good resource for identifying regulatory regions for microRNA. And we have seen 83 microRNAs in HeLa cells, and this is good to study the microRNA network. And similar strategies can also be used for other type of non-coding RNA, as long as they are RNA polymerase II transcribed, and for protein coding genes, and there's a tissue-specific alternative promoters can be identified. And more importantly, same strategies can be used in other biological systems. Actually, we did two systems, and both in breast cancer cells and in HeLa cells, and both of them are giving us very meaningful biological results. Okay, and the reason I think throughout this course, this is the first time I'm talking about my own research, and I always talk about others. And the reason for this is you see ChIP-seq experiment. If you go outside to talk to other people, most people just tell you the peak finders, and they assume that is ChIP-seq. Right? That is not. That is one part of a ChIP-seq experiment, and there's many different algorithms, many different groups already studied that. And the second part, which is the RNA polymerase II part, and this part really people didn't trying to develop too much methodologies to use this type of data. I think this is one of the attempts, and not too many available, and to use the ChIP-seq data on our polymerase II and to study the gene regulation. And the third part, which we'll talk about later, which is the epigenetics part. Again, there's a lot of significant efforts needs to be done in terms of improving the computational strategies. 45 minutes. Let's take a 5 minutes break, and then we will do the epigenetics part, and after that we will leave about 1 hour to have the course review. Okay.